All right, thanks everyone. I'm Joshua Schimmer from the Piper Jaffray Biotech team. Pleased to introduce one of the companies that's actually in the process of making that transition to the clinically validated gene therapy companies, which is an elite and growing group, fortunately. We have Tim Miller, the President and Chief Executive Officer of Abeona. Tim?
Thanks for having us here and for giving us a little bit of time to kind of give an update, talks about some of our story today. We've got a lot of things that we could select from to actually talk about, but so I had to kind of pare things down, give us a little bit of Q&A as well. So we are Abeona Therapeutics. We are a rare disease gene therapy company. And what's important is that we have a lot of products in our pipeline. One of the things that I think kind of sets us apart is just we have 3 clinical programs right now. Looking at this at our pipeline slide, the top program, ABO-102, is for our MPS IIIA program, which has treated 6 patients to date. And you can see by the check marks over on the other side of the box that we have a lot of the orphan product designations and including fast track. So we hope to be able to file that rolling BLA at some point in the future, not too far from now. And again, hope as we look to MPS IIIB, our ABO-101 program, which is going to be enrolling patients sometime in the next few months in the United States. So, and then of course our EB program, our recessive dystrophic epidermolysis bullosa program, which is another cell therapy, gene therapy play. So why we kind of wanted to update everyone here is to talk about what type of data we're seeing right now. With these lysosomal storage diseases, these kids can't produce an enzyme to break down sugars. And right now we've been able to read out the low-dose cohort data where we're actually able to see a change of 60% in reduction of the cerebrospinal fluid GAGs, which if you look around at the space of some of the other programs is really remarkable, especially when you start to layer in some of the organ changes and now neurocognitive changes. So again, looking at liver and spleen volume reductions, being able to see changes in total urinary GAGs, when you start thinking of that triangle of efficacy and registratable endpoints, looking at changes in biomarkers, changes in organ manifestations, and changes in neurocognitive effects. So we're already starting to see those only just in the low dose. Looking forward to updating on the high dose coming out data in a few months. And now as we expanded our clinical trials now to include the United States, Spain, and Australia, we're able to speed up our enrollments as many of the gene therapy programs do serial enrollments. We'll be doing that now on a per country basis and looking forward to getting 2 to 3 patients enrolling a month throughout the summertime. And really as we start to look at how we assess efficacy and start to treat more lysosomal storage diseases in this space, it's important to really kind of focus on how we're looking at measuring efficacy. Right, so Josh, I thought this also might be a nice touch point for us is, with the first couple of patients that we enrolled were slightly older, where we were hoping to see, some form of disease attenuation or disease stabilization en route to a potential reversal. Now as we shift focus now to patients that are younger, represented by the orange dots here, really these are patients that as we treat them earlier, we're hoping to continue their development plan, really as they continue to develop potentially more normally. So it's an exciting time for us as we looked to bring more and more patients on and start to read out more data. And again, we can talk a little bit more about our epidermolysis bullosa program or some of the other Batten disease programs. For those of you that haven't seen yet, BioMarin did get their CLN2 approved today. So we have 2 programs in the Batten disease space, a CLN1 and a CLN3 program, which are using the same types of gene therapies. And now as we see many of these gene therapies starting to really demonstrate efficacy, the AAV gene therapies, and looking at Spark, looking at Avexis, Abeona's programs lie very similar to those as well. So I'll kind of wrap up there. We can kind of take some questions.
Terrific. So Sanfilippo 3A and 3B have both been elusive targets, particularly the CNS aspect. Maybe you can kind of compare and contrast some of the efforts that have been pursued historically and how your strategy for 102 differs.
Sure. So many of those programs were enzyme replacement therapies, and the challenge with many of those therapies is that the enzyme doesn't cross into the blood-brain barrier. So you have to either do a direct CNS delivery to try and again show some level of efficacy, but while they're able to show increases in plasma enzyme activity or reductions in some sugars there, I think the challenge is really showing anything else, especially over a longer period of time. And going in as a single intravenous injection compared to an invasive route, drilling holes into the skull or, trying to put a device in, I think, some of the things that separate these programs.
How do we think about the natural history, both in terms of the current status of the databases for the Sanfilippo diseases, and relative to what we may know about the natural history as we think about the neurocognitive status and neuroperformance status? What are you seeing in the patients treated, and how do you think about the potential to separate from what that natural history may be?
Yeah, well, the natural history studies are really, one of the key things with many of these rare disease programs, right? Because, you need to have some form of comparator arm, but it's not necessarily ethical to treat, in a blinded, randomized, controlled manner. So we have a 25-patient natural history study that went through 1 year worth of follow-up. Really what this helped us do was validate all of the outcome measures for the clinical trial. The FDA was involved in the design of the natural history study. It was funded by foundations from around the world that believed that these gene therapies, these AAV gene therapies, are really the best route for treating their kids. So the natural history study helped us evaluate the neurocognitive, what assessments we'd be able to do, but more importantly, what window they needed to be in for us to be able to show some level of benefit. As I think we've discussed before, 70% of these kids don't reach the age 18, but they fall off a cliff developmentally between the ages of about 4 and 7. So what we're able to, I think, do now is show some level of disease stabilization, slow down their decline, and now as we move to treating earlier patients, younger patients being able to continue and show potentially some form of more normal development.
And what might we know either from preclinical models or any clinical assessment to try to correlate the benefit in the CSF gags to the brain where the pathology is?
Yeah, so the reductions in the heparan sulfate, particularly in the CSF, is really the holy grail, right? You're trying to make sure that you're using a virus that can cross into the blood-brain barrier. That's really the proof of concept that's, I think, is the one that's going to really win the day here, because when you can get across into the CNS, get into the CSF, and show those reductions, the implication is that by removing that underlying storage pathology, that's going to have a level of improved neurocognition.
And how do you, as you move upstream with the objective of identifying patients earlier and having a greater clinical benefit, how do you find those patients? Do we have newborn screening efforts, or where are we in terms of diagnosing them at the age of 1, 2, or 3?
Yeah, so for many of these diseases, there are no newborn screening kits, right? The FDA just recently approved the first kit to be able to treat or diagnose, I think, 7 different diseases, including, for example, MPS I or Pompe. Many of the states have no newborn screening in place for them. However, they actually did have a law, and 13 of them, that said that, well, we actually have to screen for this. But since there was no kits, there was a catch-22. Now that the FDA has approved the first commercial kit, these states will now be doing newborn screening. And so I think that many of the foundations that are focused on these diseases are going to be looking at developing more newborn screening kits. And as more of these therapies, as they get closer to approval, you start to see more diagnosis, and then there's a push more for newborn screening.
I guess we had a very interesting discussion on pricing models. A little bit early for you to probably be planning that far in advance, but it's got to be something on your mind. And,
sure.
And how do you see that playing out from a Sanfilippo perspective?
Yeah, this is actually a very active discussion topic yesterday at the Cell Gene Therapy Conference as well. So there's lots of models that have been proposed out there, whether you're looking at annuities or a single upfront or pay for performance. The challenge, I think, is that for a disease like hemophilia, where you can take some blood and you can assess factor IX expression, you have to only get above a certain threshold for you to be able to see a benefit. That's very easy. It also doesn't take into account the age of the patient or the disease progression. So the challenge, I think, is when looking at a muscular dystrophy, for example, or potentially diseases with a profound central nervous system, how do you evaluate that from a pricing? How do you look at maybe an 8 or 9-year-old that has severe disease progression but still wants to have access to the drug versus a 1 or 2-year-old who's very early in their disease progression? Having that same type of annuity model or a pay for performance is going to be much harder to assess. Same thing for a muscular dystrophy. If somebody that has much more severe disease progression, they may not be able to walk far enough to be able to assess that. And then you start talking about how you can game the system, right? If I was a hemophilia patient and I thought about, well, we're going to do this annuity model, what would stop me from going and giving a transfusion the day before I had to go in for my yearly assessment to see if I was going to have to pay? So I think there's a lot to be worked out still on that. And again, when you start looking at CMS and Medicare about what their hospital systems are built around, and they're not necessarily set up for an annuity for these types of gene therapies.
Why don't we shift to the epidermolysis bullosa
Butterfly skin syndrome, sure. Yeah, it's
indication,
easier, right?
and take us through that unmet need, and you had a recent data update as well.
Yeah, so butterfly skin syndrome is probably one of the worst diseases you may have never heard of. These kids, they have an underlying deficit in collagen, and it's the zipper that really holds your skin onto your underlying skin stroma. And so, if you bump their skin, the skin blisters and basically bubbles and falls off. And they spend, most of their life in severe pain, wrapped in bandages. They have to have severe treatments or baths in Bactine, every other day. So they can spend upwards of $250,000 a year just in bandages alone, right? So, when you think about how you approach some of these therapies, and again, this also gets into pricing at some point, we've been able to show our EB-101 program. We have 7 patients that have been treated. The first 4 of them are actually up through 2 years worth of follow-up, where they've shown greater than 50% wound closure, on the wounds that have been treated with EB-101. That's really remarkable when you consider that some of the other programs that have been out there, other products like Apligraft or Dermagraft, when those have actually been applied to these patients, they fall off in a month. So it's actually data that's being read out tomorrow, actually, out in Portland at the Society for Investigative Dermatology Conference. But again, it's really remarkable. There's no other approved therapies for this, and we have 7 patients treated to date.
So how does that model work, I guess, as lesions appear, they get treated until eventually they've been slowly whole body grafted? Is that the idea?
That's one of the nice things about it is you can also do repeat administrations. So each EB-101 is an administration. So you'd imagine when somebody first gets treated, they could come in for 10 different applications, and then as throughout their life, as more needs occur, you can retreat.
I mean, given the dramatic nature of what you've been able to show so far, what are the next steps, and how quickly can that move to commercialization?
Yeah, I look forward to providing an update on that in about a month after we have a few regulatory discussions coming up. So
how many patients treated?
To date, 7.
And what is the addressable patient population?
There's about 2,000 to 2,500 in the United States alone, which is the most severe form. There are about 30,000 EB patients in the United States.
And I'll pause, see if there are any questions from investors. And if not, maybe share some details on the Batten programs that you had mentioned.
Yeah, it's certainly very interesting to watch BioMarin already go through this with their CLN2 version. So Batten disease is another lysosomal storage disease. There are many different forms of it. We're looking at the infantile version and the juvenile version, so different gene products, so different drug products as well. What's nice about it is that we're using the same AAV delivery strategy for a very similar type of lysosomal storage disease. So again, as we see many of the other therapies that are coming up or already in clinical trials, Avexis is a great example. So they have 15 patients, spinal muscular atrophy. Abeona's products are very, very similar.
I guess there's been a little controversy in terms of the optimal delivery route for the CNS. Some are approaching direct delivery to the brain, others intrathecal, and you're going systemic. Maybe you can discuss the advantages or even disadvantages of systemic approach.
Gene therapy is all about delivery, right? And so the field's moved along over the past 20 years to really get to the point where showing nice specificity of using the right vector for the right disease, deliver it the right way, there may be a couple of different ways to deliver that. We are an intravenous delivery, so we can treat the whole body. Many of the diseases, I think, that show an intrathecal delivery route, we're going to end up missing treatment of the rest of the body, for example. And, I think you see more and more companies moving out of the intrathecal delivery more into, intravenous or some other route of delivery for efficacy.
In terms of manufacturing scale-up for systemic doses, what's the current status and what are the next steps there?
So we have a variety of manufacturers that we're working with per program. All of them are phase 1-2 GMP compliant, working down the path of commercial manufacturing. That's, I think, one of the challenges in any of the AAV gene therapies is finding the appropriate manufacturers to make enough of it. And so Abeona is moving down that same path too.
And is that likely to be a gaining step to commercialization?
I think there are a couple of programs where you'd probably point to right now and say that, it is a gating step. Once you get into clinical data, the CMC issues are really going to rule the day.
Got it. Any last questions? And
15 seconds. Anybody?
If not, we'll wrap up and we'll start with Adverum in just a moment. Thank you, Tim.
Thanks. Thanks, everyone. Thank you.